The IMA is proud to announce the publication of a journal article in the general science journal Helium. It is titled Breaking the Silence, Addressing Post-Vaccination Syndrome. The importance of this work lies in its plan for addressing the concerns of those affected by vaccine injury. A significant barrier to proper care is that post-vaccination syndrome is not yet an official diagnosis, which limits avenues for research, treatment, and support. Establishing this diagnosis is essential for progress. Defining PACVS. For chronic vaccine injury, the term post-acute COVID-19 vaccination syndrome, or PACVS, is gaining traction. Like long COVID, PACVS requires clear definitions of symptom clusters and clinical presentations, as treatment may vary depending on these factors. Currently, PACVS is often described similarly to chronic fatigue syndrome, but with vaccine exposure as the causative factor. Diagnostic challenges. A major challenge is distinguishing PACVS from other conditions. Our review compares biomarker alterations in PACVS and long COVID. While we are not the first to do this, it remains crucial to develop diagnostic markers that can determine causation. Even the presence of spike protein is not definitive, as recent research, for example the Yale Listen study, found many PACVS tested negative for spike protein. This does depend on the sampling and analysis methods used, however. Surveys, including those by the Yale Listen Group and our own Vitae survey, highlight the severity and debility caused by PACVS. Given that this condition is unprecedented, emerging only since late 2020, there is an urgent need for NIH and other agencies to invest in research. Our research is currently focused on several key questions. Number 1, susceptibility. Why are some individuals affected by vaccine injuries while others are not? There are hypotheses and some genetic data on susceptibility to both long COVID and acute COVID, but more research is needed on individual risk factors and differences in susceptibility for post-vaccination syndrome. The other research priority is the persistence of spike protein and mRNA. Studies by McKernan, Speaker, Koenig, and others have shown DNA contamination in vaccine vials, which may explain persistent spike protein. Additionally, the use of N1-methyl pseudouridine in the vaccine's mRNA is highly novel and rare in nature, raising further biological questions. Thirdly, diagnostics and treatment. From a practical standpoint, improving diagnostics and treatment is paramount. PACVS patients currently face a fragmented and sometimes predatory medical environment. Sharing clinical and scientific knowledge is essential for developing evidence-based treatment guidelines. We commend Dr. McCullough and his team for his contributions in this area, as well as all the others who are publishing on the treatment of post-acute vaccination syndrome. While randomized clinical trials can be slow and sometimes restrictive, clinical evaluation of therapies remains important. We are organizing a clinical trial to assess treatments for vaccine injury, which is registered and awaiting IRB approval. This work will inform our treatment guides and further research. We are also preparing a comprehensive review comparing COVID-19, long COVID, PACVS, and chronic fatigue syndrome. Treating PACVS is a top research priority, and we are committed to providing informed guidance and advancing understanding of its mechanisms. Thank you.